So thank you all for tuning in to today's webinar. Today, we're very excited to be hearing from Dr. Robert Bowser about how biomarkers support ALS clinical trials, including how they can support earlier diagnosis, better understanding of drug effectiveness in clinical trials, and prediction of disease progression. Thank you to the rest of my team for working behind the scenes to make this event happen. And a special welcome and thank you to our speaker, Dr. Bowser, who I'll introduce momentarily. My name is Tanner Dagdalen. I'm the co-founder and chief executive officer of Unite Genomics. Through the technologies that we're developing at Unite, we aim to rapidly increase the research community's understanding of ALS, get more patients into trials faster, and cut years off of the discovery cycle time for researchers. We envision a world where all patients are empowered to improve their health and no patient's disease goes untreated. And this world is 100% possible. And as a community, we can make it happen. So a little bit about this event and what to expect. Dr. Bowser will first be presenting for about 30 to 45 minutes about the current research underway regarding biomarkers in ALS clinical trials, after which the two of us will have a follow-up discussion and answer as many of your questions as possible. So if you have a question at any time during the event, please use the Q&A feature to submit your questions and we'll answer them at the end of the session. So now, without further ado, let me introduce our speaker. Dr. Robert Bowser is the Chief Scientific Officer, the John and Betty Vandenberg Chair of Neuromuscular Disease, and Professor and Chair of the Department of Neurobiology at Barrow Neurological Institute and St. Joseph's Hospital and Medical Center. He is an internationally recognized leader in ALS research, contributing pioneering efforts to discover and validate biomarkers for ALS and other neurodegenerative diseases. So, Dr. Bowser, thank you for joining us today. If you're ready, you can go ahead and start your presentation.
Fantastic. Thank you so much, Tanner. Okay, so hopefully everyone can see my screen now.
Yep, we are good.
Wonderful. All right, so briefly, again, just want to thank Tanner the opportunity to present to you today. And up now are just a couple of disclosures, incredibly relevant for today's talk, other than maybe Iron Horse Diagnostics, which is one of the companies I started that's working on biomarkers related to ALS and other diseases. Really, the presentation outline, talk a little bit about biomarkers in general, how can they be used in drug development and clinical trials. And then I'm going to go through a few of the recent clinical trials that have incorporated biomarkers, or sometimes have looked after the fact at biomarkers in samples. And a little bit on some digital biomarkers that are quite hot now and actually I think could be quite valuable moving forward. And obviously some upcoming directions for the use of markers in ALS. Not going to spend any time on this slide. I think we all know about this disease. Really, the take homes from this are, the time for diagnosis and the variable rates of disease progression, which really make it a very heterogeneous disease population that we're trying to enroll in trials. So if you look at, obviously, some of the obstacles in ALS clinical trials, we obviously have a poor track record in getting drugs through the clinic, through regulatory development, through phase 3 and onto the market. And of course, one of the big reasons is this heterogeneous patient population. There are a large number of genes implicated in ALS, either causative or risk factors of the disease. Lots of different cell signaling pathways are altered in the disease, and this variable rate of disease progression from patient to patient. All that make properly powering a clinical trial that might be less than a year of duration in order to see a signal quite challenging. Another big question that we often are faced with in clinical trials is really, does the drug actually get into the nervous system and does it hit its target? Is it doing what we want it to do? So many trials in the past actually haven't answered that question. And so at the end of the day, we're left with not only a failure in a clinical trial in ALS, but we're left without the knowledge of whether or not the drug was actually where it should be in the body and if it's actually hitting its target. Improvements are needed in how we run and manage and design clinical trials. And a big advancement is the platform trials that the Healy Center at MGH has really been spearheading. And so here, very nicely now, we have shared placebo groups within this format setting. So fewer and fewer patients on placebo within any one clinical trial. There's a base set of biomarkers that are going to be looked at in patient-derived blood samples during these studies. And obviously the continuous management and addition of more and more trials within the system that currently operates. So in the past, and some of you might have seen this when the Healy platform trials were being introduced and described, we run clinical trials now, we build up a huge infrastructure in order to run it, and then we tear it all down when that trial is over, and then reform it again for the next trial. And so in this type of setting, that infrastructure is maintained throughout, and again, greatly beneficial to the setting and also for the speed in which trials can be initiated. Finally, we need new and improved outcome measures to really help find and establish the efficacy of a disease-modifying therapy. So can we find something and have demonstration using biomarkers or new outcome measures, whether or not a drug is truly effective? So some of the goals for ALS biomarkers that I've been working on for many years now, some for an aid for earlier diagnosis, and that could be genetics. Obviously, if you have a particular genetic alteration that might predispose you to develop ALS. Protein signatures, microRNAs, metabolitic types of signatures that might aid a neurologist into diagnosing someone quicker with ALS, but also potentially to help inform and predict the course of disease within the individual. Using biomarkers to enrich patients. So this is really key, I think, for future clinical trials. We want a more homogeneous patient population in our trials. And so if we can enrich for a subset of patients that we think may best respond to a particular drug treatment, then the chances of success, the chances of actually answering the question, does this drug work in ALS or not, will be greatly magnified. And so this trying to generate and enrich for a particular patient population, I think, is going to be key moving forward. We need to better monitor the impact of treatments and during clinical trials. So now we're truly on a regulatory side, you're looking at survival, you're looking at change in the slope of an ALS-FRS, which may change slowly over time. And so can we use biomarkers to say whether or not the drug is doing what it actually should be doing in the patients? If the answer is yes, it's worth continuing through that clinical trial. If the answer is no, maybe it's not worth pursuing any longer. We should be pulling the plug. So the idea is, can we have faster go, no-go decision-making process? And really that's throughout the whole drug development process. So even on the preclinical stage and through early phase clinical trials, can we have a quicker, yes, we should continue, or no, let's move on to the next drug candidate. And then finally, again, improving our clinical design of trials and really using biomarkers to really determine who to treat and when to treat them. So lots of efforts across the field in ALS biomarker research. So large numbers of clinical research studies exploring classes of biomarkers in patients and controls. Big questions are how these change over time. We need these answers. If we're going to incorporate them into clinical trials, we need to know for an average ALS patient, how does this change over time? Because we won't be able to properly interpret the results of a trial unless we know that answer. Biomarkers that identify genetic subtypes, for instance, SOD1 carriers, or maybe C9 repeat expansion carriers. Identifying those patient populations because they may be treated in very specific manners for specific genetic subtypes of ALS, or maybe even treatment in a pre-symptomatic stage. So could you actually prohibit the disease from actually occurring in somebody? And finally, we need to incorporate more and more of these biomarkers in the clinical trials. I've been pushing that for quite a number of years now. And it's really nice to see that we're actually doing so. And some of the next steps are really, combining some of the biomarker types. So maybe looking at genetic forms, but maybe including some of the protein signatures that we find, and looking as sort of a analytical tools and multivariate analysis for different risk factors and predictions, and putting in new types of biomarkers like digital biomarkers, and ultimately developing, I think, a precision medicine approach for the treatment of patients or patient subtypes. And again, that requires that sort of homogeneous patient population. So one of the markers, and probably the best biochemical protein-based biomarker for ALS, are the groups of proteins called neurofilaments. My lab and others around the world have explored these for a number of years. And this is just some data from my lab looking at the CSF of ALS patients, where you have much higher levels of neurofilament versus healthy control subjects. But interestingly, if you look at either pure lower motor neuron disease, pure upper motor neuron disease, you can actually differentiate ALS from those as well. And so you can start to now tease out specific subtypes of the disease. And so again, it offers new insight and new abilities to track and monitor individual patients. And again, Iron Horse, that company I mentioned, we've developed validated assays for these that we're using in drug development and in clinical trials to monitor and look at neurofilament changes over time in individual patients. And this is just showing, again, neurofilament levels from yet another paper in either the CSF or in the blood, just showing that, yes, neurofilament, again, is higher in ALS versus non-ALS, but again, in either fast progressors, it's higher than in slow progressors. And if you have certain genetic types, again, this is C9 repeat expansion, they have some of the highest levels. And so you can start to tease out, again, subtypes of ALS based on particular proteins. And neurofilaments have been quite interesting because now, in collaboration with Michael Benatar and his group, collecting samples from patients that actually harbor genetic mutations for ALS, but are asymptomatic, so before any symptoms occur. And then him and his group has been following these patients over a course of a number of years and asking, when do symptom onset occur? And can we take biofluids, either blood or CSF, from those individuals before they had symptoms and ask, are there any biomarkers that are different in those individuals even before clinical onset of symptoms? And in this case, neurofilament proteins, again, were already elevated in patients that harbored genetic mutations that cause ALS, sometimes years before symptom onset occurred. So again, we're coming to the point where we can start to use biomarkers even in pre-symptomatic individuals. Of course, neurofilaments aren't specific for ALS. There's neurofilament increases in almost any neurologic disease because it's just a marker of injury to neurons or death of neurons. And so again, MS is higher, traumatic brain injury, not surprisingly, it's higher, stroke, other neurodegenerative diseases, it's all higher than healthy controls. So again, they can't be used really as a diagnostic for the general population, but they could be quite valuable for saying, yes, you have a problem in your central nervous system. And again, just looking across a host of different neurodegenerative diseases, they're all elevated compared to control, but ALS patients, it's a wide distribution, but overall higher levels than other neurodegenerative diseases. And it's not just biomarkers for neuronal injury. This is a group of biomarkers for inflammation. And so it's inflammatory cytokines, both in the blood and the cerebral spinal fluid. And again, I'm not going to go into the minutia, but certain of these cytokines actually are higher and are more elevated in people that are fast progressors versus slow progressors. Or again, these are C9 patients, they're higher in C9 patients. And we can see that in the CSF and for some of them in the blood as well. And so you have these different types of biomarkers that are all marking different types of mechanisms that are related or informative for ALS. So can we use any of these then in clinical trial arenas? So now I'm going to give you an example of one trial where we did exactly this. And this actually it's just been accepted for publication. So it hasn't been out there yet. But this is using inflammatory biomarkers in a small clinical trial for this drug called Soluble. And really, again, it was a small trial. It's a small phase 2 trial, and we're really looking for safety and tolerability of this drug in ALS patients. So this drug is FDA approved for rheumatoid arthritis. And here we know what pathway this drug hits, and it hits an inflammatory pathway called IL-6. And so the idea was, we knew from the literature that some ALS patients have elevated levels of IL-6 in their blood and CSF. And so the idea that we had was, can we look in ALS patients, identify individuals that have elevated IL-6 signaling, and those are the patients we're going to enroll in this study. And so again, it's stratifying the patient population. It's looking for a particular subset of patients because we think this drug will be most beneficial in those that have elevated IL-6 signaling. So let's specifically go after that patient population. So this was run out of my institute by one of our neurologists. It was a multi-site study. So we had 4 other clinics involved. And so again, it's looking at these inflammatory cytokines. We're looking at IL-6 signaling because it contributes in this shift of a particular type of glial cell, the microglia, into sort of a more friendly to a more unfriendly form of that cell type. Again, this is the drug. Its trade name is Actemra. And so it's a monoclonal antibody that hits the receptor for IL-6, essentially turning off its signaling. So you're trying to reduce that inflammatory signaling. And so again, we are proposing then that the IL-6 signaling is contributing to how these microglia are becoming activated and inflamed and contributing to motor neuron death. So in our study design then, we screened around 50 people. And if you had a low inflammatory signature for ALS, you actually weren't enrolled. If you had a high inflammatory signature for IL-6 signaling, then we split that into 2 groups, those that received the drug. So we enrolled 24, so 16 received drug, 8 had placebo. And it was a very short study. So they're only on drug for 8 weeks. And this is a intravenous infusion of the drug. So it was a very short study. Again, it's mainly safety. And then there is an 8-week follow-up. We collected blood at multiple time points, and then we collected CSF both at the baseline or at the beginning when drug was being introduced, and at week 8 when after the drug was done, so the last day. And so if we're looking at our biomarkers, what happened? So one of the markers that we were following was this protein called C-Reactive Protein. It's an inflammatory protein. And so what we found was that if we looked at those that on drugs, so the black circles here that were getting tocilizumab, that soon after introduction of the drug, the CRP levels dropped. It remained low through the 8 weeks of treatment. And so this arrow here just denotes where treatment ended. And then we followed them out another 8 weeks. And you can see by the end of the 8-week follow-up period, the levels were increasing again. So again, indicating that the drug was pushing down levels of CRP in the blood of these individuals, and that once off drug, it came back up again. So it was transient, and it's due to the drug treatment. If we looked into CSF, we can actually see that there was a drop in CRP levels, even in the CNS. So this was really informative because, again, this is an infusion of the drug, so it's given into the blood, but yet we're seeing a drop in inflammation even in the central nervous system. And so we know then that we're hitting the target that we want to, and that some of this drug is actually getting into the brain and turning off inflammation within the brain. So that was very exciting. We then looked at some of the IL-6 pathways, and this is actually, your body makes not only this receptor which sits on the cells and is responding to IL-6, but it also makes a soluble version of this. And so what we saw was that after treatment, the soluble IL-6 receptor levels actually increase in patients, both in the blood and in the brain. This is the body responding to the drug. It's just creating this soluble version, which actually doesn't cause inflammation. It can actually soak up and it will bind more IL-6 that's floating around. And so since the soluble receptor levels went up, we predicted that the IL-6 signal levels would actually go up. And that's exactly what we saw. So during drug treatment, levels went up both in the blood and in the CSF. And so again, the drug was doing exactly what we wanted to do. And in patients that get rheumatoid arthritis, this is the exact same thing that they have seen. So the drug was doing exactly what it should be doing. In this case, IL-6 signaling is actually inhibited, even though there's more of it around because it's binding that soluble receptor, and the drug is actually binding to the cell surface receptor. And so there's no signaling that's occurring inside the cell. So again, it was a small study. What did we find? Well, it was safe and well tolerated from ALS patients. It was not designed to actually look for clinical benefit. Again, this is a safety study. But the interesting thing was, we used biomarkers to enrich for a very specific patient population. And we found that the drug treatment did exactly to those biomarkers what we expected it to do. And so we have now evidence that we know what we should be looking for. We should be enriching for patients for IL-6, elevated IL-6, elevated CRP, if we want to go on and move this study or move this drug into a much larger study. And then interestingly enough, the IL-6 receptor actually has a genetic variant that's out in the general population. And about 25% of the population has a slightly different genotype within the IL-6 signaling. And then when we broke that out in our patient population, sure enough, those with a very specific genotype actually had the biggest impact of the drug on the biomarkers. And so it might be another way to further enrich patients in subsequent clinical trials. So that was very exciting. And again, perfect use of how biomarkers can be used in clinical trials to enrich for patients that we think will best benefit from a particular drug. I've already mentioned over 30 genes that are linked to ALS, either causative or risk factors of the disease. Some of the biggest, obviously, are the C9orf72. So it's a repeat expansion in the C9 gene, and that's in about 40% of the familial ALS patients. SOD1 was obviously the first genetic mutation identified that causes ALS. And then another gene that has another repeat expansion called ATXN2, which is actually a risk factor for ALS. And so these three genes and their genetic alterations are now been targeted for different types of therapeutic approaches. So now it's actually targeting genes for therapy for ALS. And again, using SOD1, we know that there's that the mutations cause a toxic gain of function. Almost any mutation in SOD1 will cause ALS. This is a gene that's expressed in every cell of our body, but for some reason, motor neurons then die when it's expressed over time within the individuals. So the idea was, can we lower levels? Can we just get rid of the production of this mutant form? A, will the cell survive? And then B, can we stop or halt progression? With C9orf72, again, the idea was, well, can we eliminate somehow the repeat expansion? So maybe that'll help, again, reduce the toxicity that's causing motor neurons to die. And similarly with ATXN2, it's a risk factor. So we see that in the sporadic patient population. So the idea, biologically, ATXN2 helps to modulate TDP-43 aggregation. So again, this is a protein that when we look in tissue from patients that have died from ALS, about 97% of them have inclusions of this protein. So it's a pathologic hallmark of the disease. It's also present as inclusions in patients with frontotemporal dementia and even Alzheimer's disease. So TDP-43, again, are detected in the vast majority of ALS patients. And so could we use modulations of ATXN2 and even the sporadic patient population as a treatment? So how are we going to target these genes for treatment? And the biggest approach now, and many of you have probably already heard of this, is called antisense oligonucleotides. And so we're going to use these, what abbreviated just ASOs, as a treatment. And for SOD1, Again, we're looking at knocking down the expression of SOD1 within the cells of our body. And again, how we link it in biomarkers. Well, we can measure SOD1 levels in cerebral spinal fluid or in blood. And so we know that we have a measurement within an individual. And if we add in that antisense oligo, if it's actually hitting its target, then we should see a reduction in the levels of SOD1 in your biofluids. So we can use that to monitor whether or not it's doing what it should be doing. With C9, again, it's the most common genetic form of ALS and FTD. It's this repeat expansion. So, and it causes a number of things that happen inside the cell. I'm not going to get into the cell biology of it, but within the nucleus of the cells, it causes these things called RNA foci to form. And the repeat expansion actually gets translated by the cell into forming what are called dipeptide repeat proteins, or DPRs. And some of these are toxic to the cell. And so can we somehow use ASOs to knock down the levels of DPRs? And for ATXN2, again, it's this repeat expansion within ATXN2 that's a risk factor. ATXN2 functions in how normal stress response is. So again, can we modulate this repeat expansion and maybe knock out the repeat expansion or reduce levels of ATXN2 in order to reduce the TDP-43 pathology in subsequent cell death? So real quickly, not going to go into the specifics, but obviously here's your DNA. You have a gene, it gets transcribed into a messenger RNA, and then that goes out into the cytoplasm and gets translated by these structures called ribosomes into all the proteins within our body by linking together amino acids that are encoded by the messenger RNA. In ASOs, what do? So these antisense oligonucleotides, they target specific sequences of the messenger RNA. And the overall purpose then is to regulate how much of these proteins are being made from that RNA. And it does so you add in the ASO, and it either binds directly onto the messenger RNA, and when you do that, the cell identifies and recognizes this and just degrades it. So it gets rid of it. And so now the messenger RNA for that particular gene is gone, and so it doesn't make the protein. Or you can target it so it doesn't make the protein at the level of the ribosome. Or for some of the repeat expansions, you can actually go in and modify the messenger RNA and essentially splice out or eliminate that repeat expansion. So in the world of ALS, people have either been looking at, hey, can we just get rid of the messenger RNA with these ASOs, or can we change a splicing pattern and get rid of the repeat expansion right out of the messenger RNA? These ASOs are typically delivered either in the periphery, i.e. the blood, or directly into the CNS, so the intrathecal delivery directly to the CSF. Obviously, for this, the ASOs can work for a few months, but you'll have to get repeat delivery of the ASOs over the course of one's lifetime in order to maintain the therapeutic benefit. So there is a downside, constantly being repeat delivery of these ASOs. And so for SOD1 and ATXN2, this is the area that it's been targeting. And for C9, it's really looking at this splicing. Can we splice out the repeat expansion? And this is actually some work I did a number of years ago now, but again, can we use SOD1 levels, in this case in the CSF, as a biomarker for if the ASO is doing its function or not? So the first question was, what's the level of SOD1 in an ALS patient, and does that change over time? And so what we saw, this is just a group of patients over the course of a few years. And really, once we knew the level of any individual patient, the level doesn't really change much over time. It stays constant. So that was great. So now we know if we added in the ASO, could we see a drop over time, which would be indicative of the drug having a positive impact. So now let's look at the data from the Biogen study that was published last summer in the New England Journal of Medicine. And here they're looking at, and they're measuring SOD1 levels in the CSF and looking at their treatment groups, either placebo or 4 different treatment arms of their study. And if you look at either placebo or the lowest level, there isn't really much of a change over time. And as you increase the dose of the ASO, you saw a decrease in the levels of CSF, or levels of SOD1 within the CSF, indicating then that this ASO is actually doing exactly what it should be doing, is lowering levels in expression of SOD1 within the cells within the central nervous system. If you look at the impact of it over time, measuring, ALS-FRS, slow vital capacity, so respiratory measures, handheld dynamometry, so looking at muscle strength. Again, one saw an increase or a change in a predicted way, usually within the best in signal in the fast progressing subgroup, but the more or higher dose of the ASO was the more beneficial effect to the patient. So in this case, either slowing the rate of decline of ALS-FRS, slowing the rate of decline of respiratory function, or maintaining muscle strength. And so all great clinical measures of impact of the drug. And then they also looked at another biomarker, and I mentioned neurofilament already. And so if you look at neurofilament levels, either neurofilament heavy chain or light chain, again, one saw that at higher doses of the drug, neurofilament levels dropped. Again, another indicator that motor neurons are surviving, that there's less neuronal injury and death. So another biomarker indicating that the drug is having an impact on the disease. So in this case, you have a clinical trial which showed that the drug did what it was supposed to do to SOD1 levels. So that's called a pharmacodynamic biomarker. And so it did what it should be doing. It's hitting its target. It had changes on clinical courses, which is fantastic, as outcome measures. And then it also changed a specific biomarker with respect to neuronal injury and death. So basically hit all the targets that we needed to do to help demonstrate efficacy of a drug in a large clinical trial. For C9 repeat expansion, again, ASOs are in development for this disease. And what we're trying to do, again, they're looking at, so again, the repeat expansion, which is occurring at this area within the gene. And the root cause of that, there's about 3 different mechanisms on how motor neurons might die. One of them is the generation of these dipeptide repeat proteins that I mentioned out in the cytoplasm. And the idea is we can measure these dipeptide repeats in biofluids. And so if we add the ASO, can we knock out or reduce the expression of these DPRs within a biofluid? Back to what we did initially with SOD1. Well, what happens normally over time for one of these DPR proteins in ALS patients? And again, what we saw was that individual patients had different levels, but pretty much over time, levels stayed fairly constant. And so again, this was good. If we added the ASO, then we should see a reduction of these dipeptide repeat proteins within the CSF. So we tried this in an animal model for the disease. And what we could see is exactly what we predicted, that in animals that had this repeat expansion, so this is a 66 repeat expansion, this is either looking in the brain or in the cerebral spinal fluid. If in this case, animals were treated with the antisense oligo, the C9, or in this case, a placebo group was treated with just a saline solution. If they were treated with saline, we didn't see any change over time, and we saw the DPR proteins present in both the brain and the CSF. And in the presence of the ASO, that we saw this great reduction. And so again, showing that this could be useful as a pharmacodynamic biomarker for the treatment of ALS patients with this class of drug for antisense treatment for C9 repeat expansions. So exciting work, and that is moving forward. With ATXN2, again, this is just some animal studies done with Aaron Gittler's group at Stanford. But again, now I mentioned that it was linked to TDP-43 pathology. And so they used a transgenic mouse model that has TDP-43 inclusions and showed that by an antisense oligo to reduce the levels of ATXN2, one got survival of the animals and improved motor function within the animals that are treated with the ASOs. So again, good preclinical data to support the idea that this should be moved forward in the clinical trials. Now, another way that one could treat even some of these genetic forms is using another sort of gene therapy approach, using a retroviral and adeno-associated viral vector in order to go in and provide gene therapy approaches. And Bob Brown at University of Massachusetts has been trying this in SOD1 patients and using what's called AAV vectors to basically reduce the levels of SOD1 in cells using these vectors that contain specific microRNAs that will downregulate the amount of SOD1 that's being expressed in the cell. The plus side of this approach, I mentioned the ASOs have to be delivered over and over and over again. This AAV vector can be delivered once, and then your treatment's done. So it potentially could be a life once per your lifetime type of treatment. There's still work to be done to make sure we can get long-term expression for the lifetime of an individual, but the data to date would support that idea. And so their group went and took essentially just 2 patients. It was a very small trial using this adeno-associated virus with this microRNA that targets SOD1 expression to ask, can we reduce SOD1? So again, they only had 2 patients. And this is just some of the data from clinical measures looking at respiratory function, again, slow vital capacity. So patient 1 was a more rapidly declining patient. And even after treatment, there was no change in that course. So continued on a downward trajectory. Patient 2, yeah, it looks great. There's no decrease over time in slow vital capacity, but this is probably a slow progressor. There wasn't any decrease even before treatment. And again, looking at ALS-FRS, patient 1, again, continued on a downward trajectory, whereas patient 2 remained fairly constant over time with ALS-FRS. So, we can't really say that this treatment, this AAV treatment, provided any clinical improvement or changes to the patients. And again, it's only an N of 2 here, but one of these patients unfortunately passed away. So patient 1 passed away, and they collected tissues from the individual postmortem. And then they looked and said, well, did we actually reduce SOD1 levels in the tissue itself? And what you see here is this patient 1, and looking across other patients that harbor genetic mutations in SOD1 or in just healthy non-ALS individuals where they have brain tissue, that there's a great decline in the amount of SOD1 protein within the brain tissue. In this case, was in the lumbar spinal cord. And then they asked, well, what about activity of SOD1? And again, it reduced the activity of SOD1 that remained. And so at least post-mortem measurements of SOD1 would indicate that while the AAV did not appear to change the clinical course, it did do what it was supposed to do, and that is it reduced levels of SOD1, both at a protein and activity level. Again, further work's really needed to delve into how this viral vector may be doing its job, and if further studies are needed for this particular approach at reducing SOD1 levels. Another clinical trial of interest to ALS patients is the NeuroOwn study. And in a phase 2 study that came out almost 2 years ago now, again, it was a small safety study. They looked at a couple of biomarkers for neurotrophic factors and inflammatory biomarkers in that study. And they got some interesting results. And that moved on to a larger phase 3 study, which, at the end of last year was reported that it failed. So it did not meet the clinical endpoints to demonstrate efficacy of NurOwn. Now, there's still a lot of work going on within the company looking at biomarkers in those samples, and that information, I'm sure, will come out soon. But in their phase 2 study, the cells that they were transplanting in should be creating neurotrophic factors, so things that neurons like to see and helps keep them alive. And they were hoping to get reductions in inflammation. And so they looked at 2 time points. V5 is when they transplanted the cells, and V6, they collected CSF just 2 weeks later. And at the top, looking at these neurotrophic factors, so VEGF increases upon transplantation of the cells. HGF, another trophic factor, and LIF, another one, also increase after transplantation of the cells. And in the placebo group, there's no change. And looking at inflammatory markers, again, there's an overall reduction in inflammatory markers within the treatment group and no change in the placebo group. So in this earlier phase 2 study, at least they had some biomarker data suggesting that they're getting the impact or treatment effects that they would hope to see. Again, with NurOwn, this is it requires multiple transplantations over time. So there are other challenges with that approach. But again, it'll be interesting to look at additional biomarker data when that becomes available. So trying to wrap up, a number of companies are actually looking at speech in ALS patients, and some of you might be contributing to some of these studies. A couple of these, one is a company called Aural Analytics, and they've published some of their data looking at early detection and tracking of bulbar changes in ALS using at-home speech measurements using an app on your cell phone. There are a couple other companies. This is IBM. So IBM is also using their app, looking at speech analytics and detecting changes early on in ALS patients. Google is doing the same. So a number of companies really looking at speech analytics. And I think sort of a number of these at-home measures are going to become sort of some very common features of our upcoming clinical trials. Currently during COVID, when we couldn't have patients come into the clinics, it was all at home. And so it really pushed some of the efforts at home measures in order to see if collecting even more data on a more regular basis than we can get by patients coming into the clinic every 3 to 4 months, if we can really obtain some really deep speech analytics and delve very deeply into the disease process just from speech. And this is some data from an Aural Analytics study looking at about 1,500 individuals and looking at ALS patients that actually self-report they got normal speech, and then looking at healthy controls. And so the company, using their algorithms, could actually quickly say, no, even the ALS patients that think they have normal speech don't, if you actually look at and compare them to healthy individuals. And they can rather well distinguish ALS from healthy control population, even those that say that their speech is relatively normal. And then also they have a number of data looking at speech variables that can predict subscores within the ALS-FRS for either speech or even bulbar subscores. So can we collect data at home, which basically would subserve some of these measures that we normally do in the clinic? And a number of studies ongoing with both Aural Analytics and other companies looking to see and exploring the idea of speech as a new outcome measure. And a lot of work needs to be done to demonstrate that. So it's not something that'll happen quickly, but what's required is more and more clinical research studies to demonstrate the ability that at-home measures of speech or at-home measures of respiration can be used ultimately for new outcome measures in future ALS clinical trials. So in summary, yeah, we have biomarkers that we think can be aids for diagnosis, and that obviously can be genetic mutations or protein markers that be useful for monitoring treatment responses. Obviously, some of the genetic mutations, then we can select patients for specific treatment regimens, very likely antisense oligos or maybe gene therapy approaches. Can we use gene expression profiling or protein biomarkers and enrich for patients with a specific phenotype that then is targeted for clinical trial? And again, I've shown you some of that data using Actemra. Pharmacodynamic biomarkers to make sure that a drug is actually doing what it's supposed to be doing when we provide it to patients in clinical trials. And so there are a number of these that we've identified and can show we can use in order to help demonstrate target engagement. Really need to be incorporating biomarkers into every ALS clinical trial, whether it big or small. Digital biomarkers, I think, will ultimately become a new outcome measure. And I think it's these combinations of biomarkers, of genetics, proteins, and digital that ultimately will provide the most optimal panel for either patient selection or for monitoring treatment effects in trials. So we do need continued enrollment of patients into clinical research studies. So we always need participants that want to be involved in clinical research. I've listed a few studies that have been and are enrolling patients within their clinical research studies. But again, more studies are really needed. We need to truly figure out how to interpret some of the biomarker data that we're collecting in clinical trials. The inclusion of biomarker assessments into platform trials, I think, are really going to help this moving forward. We need to figure out within ALS patients, what's the normal ranges for all these biomarkers? How do they change over time? Can a change over time predict something, either in a positive or negative way within a trial? And to help use these biomarkers to really aid for a more rapid diagnosis, but also to figure out the best and optimal treatment decisions for individual patients. So in my mind, I see things like having an individual, you have clinical features and hopefully biomarkers even before diagnosis is made. But that combination of these markers and clinical features along with some genetics, by this time it could be whole genome sequencing on people, or it could be looking at particular gene variants, or maybe RNA species that might be present within an individual. Combining that with environment and lifestyle things, maybe some of these at-home measures and speech analytics, longitudinal clinical measures, and then looking again at specific markers in blood and CSF, and maybe imaging down the road. All these sort of pumped into sort of AI approaches to figure out what's going to be the best drug to take forward. How do we enrich for specific clinical trials that are looking at a particular drug, which is marking a specific mechanism? So we should be enrolling patients that we think will most benefit by that particular drug. And again, using these approaches to obviously then monitor treatment responses so that we can identify the effectiveness of a drug or any type of treatment in our upcoming clinical trials. With that, I'll stop. I'll just mention a few people in my group, and mainly collaborators either at the Barrow, at Wake Forest with our Actemra study, the Mayo Clinic, some of the C9 repeat expansion work that I showed, and at the University of Miami for the pre-symptomatic folks. Obviously, our funding sources that were required in order to do all this work. And with that, I will stop and take any questions. Thank you.
Thank you, Dr. Bowser. Fantastic presentation. I'll just remind everyone that if you have questions to ask, please use the Q&A feature to submit your questions, and we'll answer them if we can get to them. Just to kick us off here, I have a few questions myself. So Firstly, you mentioned a couple different uses of biomarkers. You mentioned that we can use biomarkers to enrich for a particular patient population, during the enrollment process to lead to a more homogeneous group within the clinical trial. You also mentioned that we can use biomarkers sort of as a secondary outcome or a pharmacodynamic biomarker. Then the last thing that you mentioned was that we can also, or at least what I've seen in clinical trials is that biomarkers are also being used as more exploratory measures, for a post hoc or a retrospective analysis.
Yes.
So that, regardless of the trial outcome, they have some data they can use to see if those biomarkers may be used in a more prospective manner in the future. Have you seen any trends over the last, 5 to 10 years of ALS research towards the development of more biomarkers outside of the new things that we're hearing about now with the digital biomarkers and that sort of thing? I mean, kind of where does the field stand in biomarker research over the past 10 years?
Yeah, great question. It's really exploded in the last decade or so. There's still a ton of discovery work and validation of biomarkers. So it is a timely sort of process in order to both discover and validate whether or not how good a biomarker is. Obviously, we want to incorporate biomarkers in clinical development or in the clinical trial process that we think are valuable. And the only way you can demonstrate how valuable the marker is to not only have like a lab or 2 labs sort of look at it and explore it in a small number of samples, but to have labs around the world really look at it, really measure it in large volumes of samples and see, does it pass the test of time? Is it really a useful marker? Can we measure it reliably, accurately, reproducible in patients or individuals, and see if it tracks or changes over time? So yeah, there's been a lot of work and a lot of ongoing work, and some of our challenges have been, finding enough longitudinal samples. And so getting, more individuals to participate in clinical research studies that collect these samples over time becomes more and more valuable, because it's easy to discover a biomarker, and there are tons of proteins, microRNAs, all types of things that are potential biomarkers. But the challenge really is going through the next steps to demonstrate the utility of that. And so I think there's a lot in the pipeline with respect to biomarkers to ALS, but there's a lot more work to be done in order to really move them into a clinical trial arena.
And you also brought in some insight into how biomarkers are often shared between different diseases. And in particular, you mentioned Alzheimer's, and it's a particularly interesting time to talk about this because of the recent Alzheimer's approval. Right? It's
Yes.
a conditional approval, as best I understand. It's a conditional approval based on some of the biomarker data that they showed. And they still have to prove kind of overall efficacy. But to what extent is the ALS research community seeing that and building that into their trial designs moving forward now? Because it could point to potential therapies in ALS being approved despite the heterogeneity we're seeing in the overall outcome measure. If there's really solid results in the biomarkers, is that something that the scientific community is looking to now?
Absolutely. And you brought up the recent Alzheimer's disease approval, which was surprising given that it failed all the primary outcome measures, which again in the ALS world sounds very familiar. And again, having some biomarkers that look rather interesting. And so I do see that as a potential path forward. And so it could be that some of our recent sort of failures might end up finding a way to get, as you mentioned, a conditional regulatory approval in order to move forward and to generate more and more data to demonstrate overall effectiveness of their drug treatment. So I do see that as a path forward. But again, often like to highlight that, having a biomarker is one thing, making sure that you can actually and reproducibly measure it and see that in a patient population is important to do, but validating that marker is really critical, I think, to being able to use that in even a sort of a regulatory arena that the FDA is looking for. So very early types of biomarkers that haven't been studied at depth, I don't think will hit that mark, but I think some of the more heavily explored acceptable biomarkers within our field will become quite valuable for those types of study subset analysis.
Great. And I have one more question before I ask a question that I ask every speaker. So, if we look at areas like, oncology and the cancer therapeutics have been developed, a lot of the newest drugs were approved based heavily on biomarker research and biomarker results. There have been some criticisms of the newer drugs that came out in the past 10 or 15 years that they look great, they show great efficacy on the biomarkers, but they don't actually have a meaningful effect on the quality of life and that sort of thing. And so are those conversations happening as well within the scientific community around okay, great, we can show that the drug is hitting its target and it's having the effect on the target that we expect, but is that effect actually meaningful for the patients? Are they actually able to maintain function for longer or restore function, that sort of thing?
Yeah, I mean, that's a great question. And there is definite discussions within our field exactly around that. And there are some trial data around different biomarkers that would be supportive, like the Biogen study, that, yeah, this is a great use of markers and helps demonstrate and tracks, and we can see not only a biomarker impact, but a clinically meaningful impact. And then we have other trials where maybe that did not happen. And so how do you interpret that? And I think our field in neurodegenerative diseases in general are probably behind the oncology space in this regard. So we don't have that wealth of years of data to look back to. And so I think we need to accumulate that information and that data in our field, but it's going to take some time. And so I think there's definitely discussions and there's going to be ongoing debates on how to interpret biomarker data within a clinical trial setting. And if you see a disconnect between the two, what does that mean? Does that mean the drug wasn't beneficial, or the biomarkers providing no meaningful information in that particular trial? And so I think that those discussions are ongoing. They're going to become a little more boisterous, I can see, in the next probably 6 to 9 months. But I think, as a field, we're sort of at the early stages of this. And, by incorporation of biomarkers in the ALS clinical trials is still relatively new.
Great. And, a question I ask every speaker here is, what can the patient community do to accelerate treatment development right now? I mean, in the context of biomarkers, you mentioned a number of longitudinal studies. I think we're going to be sending out the recording and the slides or the recording after this presentation, so everyone will have access to that and can go look up those longitudinal studies. Is there anything else that the patient community can do to accelerate the treatment development pipeline?
Yeah, really. I mean, again, it truly is participating in studies. Right. That and always looking and trying to push for additional funding sources to do this type of work. I mean, trying to do clinical research studies in order to collect the samples, it's just not a funded type of thing at the NIH level. It's really hard to get money for. But there is a really key and critical feature on how we're going to move this field forward and develop more effective therapies. And so, yes, voicing opinions regarding the value of these types of clinical research studies is really key. And then participating in the studies is critically important. And I'd be happy to help people find what studies are near them.
Wow, thank you. Well, with that, this concludes today's discussion. Thank you again for joining us, and a very special thank you to Dr. Bowser. For all of the attendees here, we'll be sending a post-event evaluation form So keep an eye out for that in your emails. And, we use that feedback to determine where we go next with these webinars. So in general, we appreciate everyone's support and we look forward to what we'll accomplish as a community in advancing ALS research and treatments in 2021. As always, the time to act is now. So let's get out there. Let's find these longitudinal research studies and get involved.
Thank you so much.
Thank you.
On board.
Take care, everyone.
Take care, everyone.